Hi everyone, I hope you all had a good weekend. So today is going to be covering quite a bit of stuff, so we might go a little bit over time. I don't mind if you guys have to leave at 8 o'clock, just letting you guys know. So first of all, we'll go over alcohol metabolism. So alcohol or ethanol provides 29 kilojoules per gram of energy. So it's worthwhile remembering that number, okay? So ethanol is 29 kilojoules per gram, and it's turned into acetaldehyde by alcohol dehydrogenase, and acetaldehyde is then turned into acetate by aldehyde dehydrogenase inside the liver cells. And so remember that these are all 2 carbon molecules. So ethanol is just this one here, becomes acetaldehyde, which is oxidized, and then that's turned into so that's ethanol, acetaldehyde, and acetate, okay? And they're all 2 carbon molecules, and that happens inside the liver cells, okay? So that's why if you drink a lot of alcohol, you can end up damaging your liver, and we'll go into a little bit more detail about that. So each of these reactions, if you look at the diagram on the bottom, the enzyme, so each of those reactions uses up a NAD plus and turns it into an NADH. So when you go from ethanol to acetaldehyde, this one's catalyzed by alcohol dehydrogenase, and then we're taking a NAD plus and turning that into an NADH. Okay. And then the second one is aldehyde dehydrogenase, and again we're going to take a NAD plus and turn that into NADH, right? And this will be acetate. Okay.
Apologies, Tom. Some of your copies were still in the photocopy, so that's why you were short. But anyway, there's a whole bunch more.
Okay, thank you.
Okay.
Cool. Cool. There you go. Sorry. Right. So if we go back up to the top, so on that third bullet point, each of these reactions produce a NADH from NAD+, so that means that 2 NADH are produced for every alcohol molecule that's metabolized. And the acetate is combined with a coenzyme A molecule to form acetyl-CoA. So this acetyl-CoA, so we're going to combine this with a coenzyme A molecule to create acetyl-CoA. And this acetyl-CoA will enter the citric acid cycle, and that will result in the production of more NADH. So, and the NADH will lead to more ATP production via the electron transport chain. And so this causes a decrease in the ADP concentration and a rise in ATP concentration inside the liver cell. So the electron transport chain is going to be inhibited since there is enough ATP. So remember that if you have the goal of the electron transport chain and the citric acid cycle is to create ATP, and if you have enough ATP, you're going to start to slow down the electron transport chain and the and your citric acid cycle. So if the electron transport chain is slowed down or inhibited, that means that NADH can't drop its electrons off, and so it's not going to be oxidized, and NAD plus is not going to be regenerated. So it cannot be oxidized to regenerate NAD plus, and that will cause an increase in the NADH over NAD plus ratio inside the liver cell. So high levels of NADH and ATP inhibit the citric acid cycle, which means that less acetyl-CoA enters the citric acid cycle, and this leads to an increase of acetyl-CoA. So I'll just fill out that diagram that we have at the bottom there. So this acetyl-CoA will go into the mitochondria. So it'll go into the mitochondria. So acetyl-CoA. So it goes through the citric acid cycle, and at the end of the citric acid cycle we get NADH, we get FADH2, right? And then these electron carriers are going to drop off their high energy electrons in the at the electron transport chain, which is on the inner mitochondrial membrane. So that's the electron transport chain, and that generates the proton motive force, which results in the production of ATP, right? And so the high levels of ATP, so ATP levels are going to go up, and this ATP will inhibit the electron transport chain, and it will also inhibit the citric acid cycle. Okay, and if the citric acid cycle is inhibited, that means that acetyl-CoA is going to increase in concentration, right? And even though acetyl-CoA is not being used up in the citric acid cycle, if you're drinking a lot of alcohol, this is still going to keep happening because your body wants to get rid of this alcohol and it wants to get rid of this acetaldehyde. So all this stuff here is going to keep happening, and it's just going to build up, and you're going to get a buildup of acetyl-CoA. So in the boxes at the bottom, you should say increased levels of NADH and ATP, and also acetyl-CoA, and decreased levels of NAD+. Okay, so we'll move on to the next page. So if we have a high concentration of NADH, the cell doesn't like that. So the cell doesn't like having high levels of NADH, and so it'll try to convert that NADH into NAD+. So we use pyruvate, and we convert pyruvate into lactic acid to regenerate NAD+. And the high concentration of lactic acid causes the blood pH to decrease. In other words, becomes more acidic. And pyruvate is one of the main substrates for gluconeogenesis. Okay, so I'll spell that out. Gluco for glucose, neo for new, and genesis to create. So we're creating new glucose, and pyruvate is the substrate for gluconeogenesis in the liver. So in this liver cell, you're converting all your pyruvate into lactate or lactic acid, right? So you've got lots and lots of NADH in the cell, so we want to get rid of it. So that's going to drive this reaction to the right to try and regenerate NAD+, and that means that the level of pyruvate in the cell goes down, and if the level of pyruvate goes down, then we are not able to convert this pyruvate into glucose. So this is gluconeogenesis, and that will be not happening. Okay. So remember that the citric acid cycle is inhibited since there's plenty of NADH being produced from the alcohol being broken down. And this means that acetyl-CoA will not be entering the citric acid cycle, so it will increase in concentration within the cell. This is a bit repetitive, but I'm just trying to get you guys to remember that because it's important. Excess acetyl-CoA results in increased fatty acid synthesis inside the liver cell. So we're creating fatty acids by putting those two carbon molecules, the acetyl-CoAs, together, and we're building up a long chain of fatty acids, and this can lead to fatty liver. So if you look at the diagram, we've got lots of acetyl-CoA. So we're going to add the acetyl-CoAs together to create fatty acids, right? And then we're going to combine it with glycerol to make triglycerides. And so if you have a lot of triglycerides being made inside your liver cell, then that's just going to build up. So some of it will be exported into the blood, but if you're constantly drinking alcohol over a long period of time, you'll end up getting a fatty liver. Okay. So we'll move on to the next page. Yep. No, fatty acid is different. So lactic acid is 3 carbons, and fatty acids are usually about 16 carbons long. So the most, yeah, it's a lot longer. So, yeah. Right. So we'll move on to the next page. We'll talk about the microsomal ethanol oxidizing system, or MEOS. So if you're consuming a high amount of alcohol all the time, we have an alternative pathway that can also break down ethanol into acetaldehyde. So this pathway is known as MEOS, and it's toxic because it results in reactive oxygen species. So reactive oxygen species, or ROS, and that can damage the cell. So chronic high consumption of alcohol can increase the activity of MEOS and result in increased breakdown of other drugs which can cause problems with medications and also results in greater production of reactive oxygen species. So that's just a way of your liver cell adapting. So if you'll notice that once you start drinking alcohol regularly, you're able to break it down faster and you develop a tolerance. So part of that is because your liver cell is upregulating it or creating more of this MEOS, which means that you can process alcohol faster. So if you look at that, this MEOS here, you've got ethanol, and that's being broken down into acetaldehyde. And this also uses an oxygen, right? And that's going to, oxygen is very electronegative, and that will cause the reactive oxygen species, which will cause damage. So you can see it causes DNA damage, which can lead to cell death or liver cancer. And it will also damage the cell membrane. And it can also break down certain types of medications, and they'll become inactive. Okay? So that's why if you're taking antibiotics, they'll tell you not to drink alcohol, because it can interfere with the metabolism of those drugs. So fat storage, it's advantageous to store energy in fat because fat is very energy dense. So it's 38 kilojoules per gram compared to 17 kilojoules per gram for protein and carbohydrates. Okay. So 17, 17, 29, 38. That's the amount of kilojoules per gram. So 17 is protein and carbohydrates, 29 is alcohol, 38 is fat. Okay. So you should try and remember that. Triacylglycerol is stored in adipose cells or fat cells where they can be mobilized. So mobilized, that just means that we're getting them out of the fat cell and getting them into the blood during periods when energy is required. So excess glucose, alcohol, and amino acids can be turned into acetyl-CoA, which can then be used to synthesize fatty acids. And then 3 of these fatty acids can be joined to a glycerol molecule to form a triacylglycerol, and this process is called lipogenesis. So lipo for fat and genesis for create. So we're creating fat. It happens in the liver, and the liver will package the triacylglycerols into VLDL particles, which enter the bloodstream. So you've got glucose, amino acids, and alcohol, and these will all be turned into acetyl-CoA. And then acetyl-CoA will be turned into fatty acids. Okay. So this process of converting acetyl-CoA into fatty acids is going to be stimulated. It's going to be stimulated by insulin, because remember that acetyl-CoA can also go into the citric acid cycle to create energy, but if we have a lot of insulin, that means that we've just eaten, so our blood sugar is going to be high, so we don't need to make any more energy. We want to save it for later. So insulin is going to stimulate the production of fatty acids, and then it will join up with glycerol to make triglycerides. And then those triglycerides will go into VLDL packages and be exported into the blood. Okay, so that's fat storage. So adipocytes. Spell that out. Adipocytes. So adipo means fat, cytes means cells. So fat cells can take up those triglycerides for storage from the VLDL and chylomicrons. The glycerol can be formed from glucose. So glycerol is a 3-carbon molecule, and glucose is a 6-carbon molecule. So if you split glucose in half, you get glycerol. There's a few more steps involved in that, but and then lipogenesis and synthesis of fatty acids. So that's this process here. Those are stimulated by insulin. Okay. And the ability of the body to store fat is essentially unlimited, as adipocytes can divide once they get to a certain size. Okay. So we'll move on to the next page, fat mobilization. The triacylglycerol in adipose cells can be hydrolyzed into 3 fatty acids and a glycerol molecule by a lipase. So what's the name of this lipase? Yep. So hormone-sensitive lipase, HSL. This is stimulated by adrenaline. So that's during exercise and sympathetic stimulation, and also glucagon, which is released when blood glucose levels are low. So remember that insulin will inhibit hormone-sensitive lipase, because if we have a lot of blood glucose, we don't want more fatty acids in the blood, right? We want to use up that glucose, not fatty acids. When blood glucose levels are low, the decreased level of insulin, so if blood glucose levels are low, we're not going to have a lot of insulin in the blood. So that will mean decreased inhibition of hormone-sensitive lipase. And that decreased inhibition means that there's increased activity of hormone-sensitive lipase. Okay. So if there's increased activity of hormone-sensitive lipase, they're going to be hydrolyzing those fatty acids, and those fatty acids are going to enter the bloodstream, where they're carried around by albumin. And they're going to be carried around by albumin in the bloodstream until they reach a cell that can break them down via beta oxidation for energy, like the heart. So the glycerol is going to enter the bloodstream as well, and it goes to the liver, and it can be turned back into glucose by gluconeogenesis. So you have to remember, this is very important, you have to remember that the brain cannot use fatty acids for energy because they can't cross the blood-brain barrier. So your brain is a very sensitive organ, and your blood-brain barrier is very, very selective. So it's not going to let fatty acids go through that barrier, because these fatty acids would cause disruption of the neurotransmission in the brain. So we have our diagram there. So if we've got triglycerides, and we have hormone-sensitive lipase turning triglycerides into glycerol and fatty acids. So this is going to be stimulated by adrenaline. Okay, so if you're running away from a tiger, you want to have enough fatty acids to keep you going for energy. And it's also going to be stimulated by glucagon. So if you have low blood sugar, then you want to have more fatty acids for energy. And it's going to be inhibited by insulin. Okay. So glycogen synthesis is called glycogenesis. That's not too important. Just remember that glycogen synthesis is stimulated by insulin. So remember, glycogen is a storage molecule of glucose. So if you want, insulin is going to be released when you have lots of glucose. So you're going to try and save the glucose for later. And in that process, you create something called UDP glucose. So just remember that. The next page, we'll talk about the mobilization. We'll talk about the mobilization of glycogen. So the breakdown of glycogen is called Glycogen Lysis. So glycogen lysis. So lysis means to break down or break. So breaking down glycogen. And that's stimulated by glucagon and adrenaline. The glucose that's produced from glycogen in the muscles will stay inside the muscle. So that's used as energy for the muscle cell. And the glucose that's produced from glycogen in the liver is released into the bloodstream. So this helps to maintain the blood glucose level in between meals. Okay. So we've got our muscle cell. Now, glycogen will be turned into glucose. And now, if we already have a lot of glucose, then we don't want to break down glycogen, so insulin will inhibit glycogen breakdown. Whereas if we're very scared and we want to run away from a tiger, then we want to have enough glucose to power our muscles. Okay? But this is just a minor thing, but just remember that glucagon does not affect the muscle cell. Okay? So glucagon doesn't affect the muscle cell because this glucose stays inside the muscle cell. It's not going to go into the bloodstream. So the role of glucagon is to raise blood sugar levels. So since this glucose just stays inside the muscle cell, there's no point for glucagon to act on the muscle cell. Now, on the other hand, we have the liver cell. So the liver cell, and again, we're going to have glycogen turned into glucose. So this time, it will be inhibited by insulin as well. And it will also be stimulated by adrenaline, and glucagon is going to stimulate the liver cell to break down glycogen. And this glucose can go into the blood and raise the blood sugar level. Okay. Right, so we'll move on to the next page. So we'll talk about the daily energy needs under normal conditions. So what I'm talking about now is under normal conditions. So the brain uses 120 grams of glucose, so 120 grams of glucose per day. That's about the same amount of sugar in 2 Powerade bottles. I think a bottle of Powerade has like 60 grams of sugar. So remember that the brain can't use fatty acids as they can't cross the blood-brain barrier. So its sole source of energy is glucose under normal conditions. And that's why it's critical to maintain a blood glucose concentration of 5 millimolar or 5 millimoles per liter, so that the brain has a constant supply of glucose. Proteins are not usually broken down for energy because they are required for organ function and maintaining the structure of your body, and also for your muscles as well. You don't want to break down your muscles. So most tissues in the body are getting their energy from fatty acids. The exception is the brain, like we talked about. And the adipose tissues, so your fat cells hold a reserve fat that can be used to provide fatty acids in between meals and while you're sleeping. And the liver stores about 120 grams of glycogen as well. So that's enough to supply the brain with glucose for 1 day, right? So if the brain uses 120 grams of glucose a day, and the liver stores 120 grams of glucose, that will last you for 1 day. So glycogen lysis, the breakdown of glycogen is stimulated by glucagon. And remember that glucagon is released when blood glucose levels fall, right? So now we'll talk about what happens during starvation. So once the liver has run out of glycogen, so after 24 hours, your liver will completely run out of glycogen. You have to start synthesizing glucose to maintain the blood glucose levels. So this takes place mostly in the liver. So it takes place mostly in the liver and a little bit in the kidneys. So glucose can be produced from mostly amino acids from muscle breakdown, and also glycerol, which comes from triglyceride breakdown. And gluconeogenesis is just the opposite of glycolysis, except that the last reaction of glycolysis, phosphoenol pyruvate or PEP to pyruvate, is irreversible. So therefore pyruvate has to be converted into oxaloacetate, which is a 4-carbon molecule first, and then into phosphoenol pyruvate. So I'll just draw that out for you. So glucose gets broken down into phosphoenol pyruvate, and then it gets turned into pyruvate. So this whole thing here is called glycolysis. But when you want to go from pyruvate to glucose, all these steps here are reversible. Oh, except there's one intermediate step here that's irreversible as well. But we'll just focus on this part for now. So this step here is irreversible, so we need to turn pyruvate into oxaloacetate, and then that can turn into phosphoenol pyruvate and go back to glucose. So if we go back up this way, that's called gluconeogenesis. Okay, so do any of you remember where we've seen oxaloacetate before? So it's part of the citric acid cycle. So if you remember the citric acid cycle, you've got acetyl-CoA, and it's joining with, so this is a 2-carbon molecule and it's joining with a 4-carbon molecule. That was oxaloacetate, and it became citrate, right? So this is the same thing here. And you'll see why that's important later on. So we'll just look at that diagram. So during starvation, cortisol. So you guys know what cortisol is, right? So cortisol is released during the stress response, and if blood glucose levels are very low, that's also a stressful situation for your body as well. So cortisol will be high during starvation. Adrenaline will be high. And glucagon will be high because you're not eating, so your blood sugar is going to be low. And because your blood sugar levels are low, you don't need to produce insulin, so your insulin will be low. So in your muscle cells, you've got proteins, so your muscles are made of proteins. And they can be broken down into amino acids. So cortisol will stimulate the breakdown of proteins, whereas insulin will prevent the breakdown of proteins. And then you have your vital organs, right? So those are things like your kidneys, your stomach, your spleen, your lungs, all those important organs. And they're also made of proteins, so that will be the same deal. So it'll just be exactly the same. You'll have cortisol, which will stimulate insulin breakdown. Cortisol, which will stimulate protein breakdown, and insulin, which will inhibit the breakdown of proteins. And then you have your adipocytes, or your fat cells. So triglycerides turning into glycerol and fatty acids. So adrenaline and glucagon are going to stimulate hormone-sensitive lipase, and insulin is going to inhibit hormone-sensitive lipase. So remember that in starvation, insulin will be low, and while cortisol and adrenaline and glucagon will be high. So you're going to have a lot of amino acids being produced, and a lot of glycerol and a lot of fatty acids being produced as well. So these amino acids will go to the liver, and they'll be turned into pyruvate, right? So remember that amino acids have an amino group on them. So we need to take those amino groups off. Oops. So what is this? Yeah, so this is ammonia. So this, we need to deaminate them. So deamination or transamination. Okay, and then we've got the glycerol. So the glycerol can be converted into pyruvate as well. So you need to remember that fatty acids cannot be turned into pyruvate. Okay, so don't make that mistake, because that's probably the most common misunderstanding I see. So fatty acids cannot be turned into pyruvate, which means that if they can't be turned into pyruvate, they can't be used for gluconeogenesis. Okay? So you can have lots and lots of fats, but those fats aren't going to be converted into glucose. Okay? So pyruvate, in order to make glucose, has to be turned into oxaloacetate first, and then it will go through the steps to create glucose. The glucose will go to the brain. Okay, and if we're starving, we don't want the muscles to be taking up the glucose, because we need to save it for the brain. So none of the glucose will be going to the muscle, because there's no insulin telling the muscles to take up the glucose. Okay. So we'll move on to the next page. So what stimulates the breakdown of proteins? Yep, cortisol. So cortisol stimulates the breakdown of proteins into amino acids, so they can be converted into glucose. Since blood glucose levels are low, there will be very low insulin. So muscles require insulin in order to express glucose transporters on their cell surface. And this means that muscles won't be able to take up any glucose, which leaves the glucose in the blood for the brain. But that also results in muscle weakness. So if you haven't eaten for a day or two, then you'll notice that you feel quite weak, because your muscles aren't able to take up any glucose. And if there's no glucose in the muscles, there's no energy for them to use. Okay. So what I'm talking about now is all under conditions of starvation, so keep that in mind. So the cortisol and the lack of insulin cause muscle breakdown, and once there's no more muscle to get protein from, then we're going to start breaking down the protein in vital organs for energy. And if too much protein is lost from these vital organs, then the result is organ failure. And if your organs fail, you're going to die. So that's pretty much, see, inside the green box should be cortisol, and then the red box will be insulin. So during starvation, insulin will be low and cortisol will be high. Okay. Yeah, and so it's pretty much the same thing, but I really want you guys to understand this. So you've got the amino acids which go to the liver. The amino acids get turned into pyruvate. The pyruvate gets turned into oxaloacetate, and then the oxaloacetate gets turned into glucose. Okay, and then the glucose from the liver goes into the bloodstream and it goes to the brain. Okay. So we'll talk about the fates of acetyl-CoA. So acetyl-CoA is like this big meeting point where the acetyl-CoA can go down several paths. So we've got acetyl-CoA. So we can turn acetyl-CoA into fatty acids. Right, and the fatty acids can then be combined with glycerol to turn into triglyceride. So when we have a lot of energy, when we've just eaten, then we're going to want to go and create fats. Okay, so if you have lots of insulin, that's going to stimulate this pathway. Okay, and also if you have no energy, so if you're starving, then this acetyl-CoA can be turned into ketone bodies. Okay, so during starvation, it gets turned into ketone bodies, and the ketone bodies will go to the brain. Okay, so we'll talk about that a bit more. And then the other way that it can go is it can combine with oxaloacetate, right? And turn and then enter the citric acid cycle. Okay. Right. So the production of ketone bodies. During starvation, blood glucose levels will be lower than normal. So glucagon will be high and it will be stimulating hormone sensitive lipase. therefore glucose will be stimulating hormone-sensitive lipase because it is high in adipose tissue to hydrolyze triacylglycerol into glycerol and free fatty acids. So the fatty acids get into the liver, and in the liver they're broken down into acetyl-CoA via beta oxidation. Right. Under normal conditions, the acetyl-CoA would combine with oxaloacetate to enter the citric acid cycle. So that would be under normal conditions. But remember that the oxaloacetate is being shunted into gluconeogenesis. So inside the cell, inside the liver cell, we don't have a lot of oxaloacetate for acetyl-CoA to combine with. So this acetyl-CoA, so that leads to an increase in acetyl-CoA. So remember when we drank a lot of alcohol, we also had an increase in acetyl-CoA. And in that situation, we turned the acetyl-CoA into fatty acids. But in this situation, where we're starving, we don't have a lot of glucose in our blood. So since the body is in starvation mode, fatty acid synthesis is going to be inhibited, right? Because if you're not eating, you don't want to be making fats. So fatty acid synthesis is inhibited. So we're not going to make fatty acids. And the acetyl-CoA will be used instead for ketogenesis, which is the production of ketone bodies. So you've got the triglycerides, which are getting broken down by hormone-sensitive lipase. And remember that you have a lot of glucagon, which is stimulating hormone-sensitive lipase. So when you're starving, you're going to get a lot of fatty acids in your blood. And then these fatty acids will be broken down into acetyl-CoA. So this process here the breakdown of fatty acids into acetyl-CoA is beta oxidation. And where does beta oxidation occur? Say it out. Yep, that's right. Well done. Mitochondria. And then, so remember that when we have, if we had a lot of energy, then this could go to fatty acids, right? But glucagon, so glucagon is going to inhibit fatty acid synthesis. So this isn't going to happen. Or acetyl-CoA could join up with oxaloacetate to go into the citric acid cycle. But we don't have a lot of oxaloacetate because most of this oxaloacetate is being turned into glucose. So instead of going down this pathway, oxaloacetate will go down this pathway. So that means that acetyl-CoA has no other option but to go down the ketogenesis pathway. So it will be converted into ketone bodies. And glucagon right? So glucagon is going to stimulate the production of ketone bodies, right? And so these ketone bodies can then travel to the brain, and these ketone bodies are able to enter the brain, and they can be broken down into acetyl-CoA. So now the brain remember that the brain was using 120 grams of glucose per day. Now it's got ketone bodies to use for energy. So we had glucose, and this glucose was being made from the amino acids that were being broken down from proteins, right? So we can now reduce the amount of protein that needs to be broken down to provide glucose for the brain. Okay. Yeah, and then so one more page. So ketone bodies are created by putting two acetyl-CoA molecules together. So we're in the liver. Got an acetyl-CoA. So putting them together, you get acetoacetate, which gets turned into, so it can be turned into beta-hydroxybutyrate. But don't worry about remembering those names. Just keep in mind that these are made from two acetyl-CoAs being put together. So remember, the acetyl-CoA is 2 carbons. So 2 carbons plus 2 carbons means this is a 4-carbon molecule. Okay. And both of these are ketone bodies. Okay. And so these will enter the bloodstream. And then enter the brain. So just, this is the brain here, big blob. And then, so once the ketone bodies enter the brain, it gets broken back down into 2 acetyl-CoAs, and then that can be, this will go into the citric acid cycle, right? And then turned into ATP. All right. And that means that we now need less glucose to be produced. Yeah. So I'll just start again from the top. Ketone bodies are created by putting 2 acetyl-CoA molecules together. They enter the bloodstream and can pass through the blood-brain barrier. They're able to do so because they're only 4 carbons long and don't disrupt neurotransmission. In the brain, they can be broken back down into acetyl-CoA. And now this means that the brain does not need as much glucose. So it only needs 50 grams of glucose per day now. So that's less than half, since it's now getting some of its energy from ketone bodies. So these ketone bodies can break down and give off something called acetone. So acetone can be exhaled in the lungs, and it gives your breath a fruity smell. Okay, so if someone comes into the hospital and they're in a coma and you can smell their breath and it smells fruity, that probably means that they're in ketoacidosis. So that's something that you look for if someone comes into hospital and in a coma. Then you know that they're probably and that usually happens from diabetes, which is something that you'll cover this week, I think. Since the brain now needs less glucose, gluconeogenesis can slow down and protein breakdown will also decrease. And the level of ketone bodies in the bloodstream increases over 100-fold or 100 times during starvation. Okay, so normally a normal person won't have a considerable amount of ketone bodies in their blood. So if you look at that diagram there, you can see that the purple line, which is glycogen will run out in 24 hours. And as glycogen is running out, we are creating more glucose. We are creating new glucose. So that's the middle line. So gluconeogenesis increases after 1 day of not eating. And then ketogenesis is also increasing. So that's the top line. And once ketogenesis is kicking in, then that means that the rate of gluconeogenesis can decrease. So that's why you see that this is the line for gluconeogenesis. It goes down. So at this time here, we're starting to have enough ketone bodies being used by the brain, and so then the rate of glucose production can decrease. Okay. So pretty much every year they give you like a 10-minute question on starvation. So they'll ask you like someone was stuck in a cupboard and explain how they were able to survive for 40 days or whatever. So it's quite good to really understand how starvation works. Okay. Yeah, thanks. Cheers. Cool. Cheers.
Cheers, Tom.
All good.